Hello everyone. I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.J. Hariharan. So today we are going to discuss about an introduction to the enumeration of bacteria. Otherwise we can also call as a method of measurement of microbial growth. Otherwise we can also tell how the number of microbial cells present in a sample can be counted. So this is a brief outline. What are the different methods available for counting a particular microorganism in a sample or counting bacteria in a sample, right? So let us consider the measurement of microbial growth. So the microbial growth is an important aspect, especially in the industrial microbiology, because while we are producing certain drugs like antibiotics or vitamins or DNA products, the number of cells is responsible for the product formation. So we have to measure how much is the cells are present in that particular growth phase of the bacteria, because most product production will be either in the log phase or in the stationary phase of the microbial growth curve. So this measurement becomes very important. So the measurement of microbial growth is generally done for two purposes. One, to determine the growth rate, or to determine the generation time, that is the doubling time. These both are essential to calculate the rate of product formation in an industrial microbiology. So whenever we talk about the growth in microbiology, it generally refers to the total population growth, not the individual growth of the particular cell. So if you see the different measurements of microbial growth, so it can be measured by three ways. One, by counting the number of microbial cell, that is the cell count technique. Second, by measuring the number of microorganisms through its weight, that is the cell mass. Otherwise, by analyzing number of microorganism by its cell activity. These are the three basic principles how the measurement is carried out. So the first one, that is the measurement by cell count. So this can be done by two ways. One is the total count. It is otherwise called as a direct method. And second is the viable count. It's an indirect method. So the term total count represents that it helps to count both the living as well as the dead bacteria or microorganism present in the sample. That means it gives a total count whether a living is viable organism are there or dead organism are there. So this can be generally done by two ways, either with the help of a microscope, otherwise with the help of an electronic particle counter, an automated machine. The second method is the viable count method. Viable means living cells. That means these cells can only develop into a colony by dividing itself. That's why it is also called as colony forming unit, the viable cells. It's an indirect method. Only the living cells can be estimated by this method. There are two common methods available in this technique. One is the plate count method and second is the membrane filtration technique. So I have given a separate lecture for the each method that is involved in the cell counting technique. The microscopic method also is available in a free video lecture. You can watch it later. And an electronic particle counter is available separately. Similarly, the viable count also. So the microscopic technique, or generally three methods are available. One is the Braid's method, second is the Wright's method, and third is the Petrov-Horstner counting chamber method. So recently the Petrov-Horstner method has been widely used for direct counting with the help of the microscopy. The electronic counters or an automated machines, generally the Coulter counter and the flow cytometers are generally used for counting this microbial cell. So it gives an immediate reading of the total counts, whereas it needs less time for analysis when compared to the microscopic technique. So the second method of measurement of the cell, number of cell, is based on the cell mass. So it is also done by two ways, a direct method as well as an indirect method. In direct method, this cell mass can be determined by simply weighing the cells, number of cells which has been isolated or centrifuged. We can weigh it and directly measure the mass of the cell. Otherwise, by proportionating the measurement of cell nitrogen with that of the cell mass. And the second method is the indirect method, which is mostly widely used in industry, that to measure the cell mass based with the help of certain instruments like spectrophotometer. The commonly used UV visible double bill spectrophotometer will help us to calculate this cell mass. So we have a separate video on this aspect also. Then a nephroturbidometer, which helps to calculate the cell mass based on that formation of the turbidity because of cell growth. So this is the another way. We have separate video lectures available in this aspects also, you can view it. And the third major method of counting the cell is the based on the measurement by cell activity. So no direct method is available. An indirect method of measurement the cell activity is to measure the biochemical activity of the cell. If the cell grows, it consumes certain nutrients. Based on the consumption of this biochemical activity, we can measure the number of cells present in that particular sample. I think this lecture will be very helpful for you for understanding and basic outline about different methods that has been used for cell counting technique, that is the enumeration of a microorganism or a bacteria. So thank you very much. So continuously view my lectures for your future updates on this enumeration of bacteria. Thank you very much.